2 was talking about a student or a scientist, and they're investigating different insecticides on the growth of a plant. And we're gonna go back over these experiments more and more, because I want to make sure you can identify the independent variables, the dependent variables, the constants, the controls, and things like that. But this happened to be on this graph. They asked you to identify where the student was wrong on the graph. So it says identify the error and provide reasoning to support why the student chose to do a logarithmic scale on the y-axis. Okay, so it's 2 parts. Okay, so the first part, let me zoom in, is where were they wrong? And they were wrong at which point? Okay, and they were wrong right here. Okay, in the immature seed / seed formation zone. This one was supposed to be around 2,000, and they graphed it around 200. So it should have been way up here, because the other one was at about 3,000. So up here, okay? And then the plus or minus is where we get these big lines, okay? So in the plus or minus on the mature seeds was a huge range. So they were all over the place. But that was where the error was. So the error is right here. Should have been more up here. And then if you notice the scale is logarithmic, because this goes from 0 to 1, and this goes from 1 to 10, so that's only 1 jump. This is 10 jumps. This is 100 jumps. This is 1,000 jumps, and this is 10,000. And normally we don't graph this way, but because the data was so spread out from very, very small amounts, was this a 0.4? Like something really tiny, 1 to 1.4, to something like up in the 10,000, like 9,490. So to draw a graph that large would be absolute ridiculousness. So you need to look out for things like that. They may give you this weird scale. Since you're not going to have to draw a graph, they may have some crazy graphing stuff for you. All right, so that was number 2. All right. So give yourself a point for identifying where they were wrong, and then give yourself a point for explaining why they spread it out so far. All right, number 3. The question they gave you, they have 3 different conditions. They have time, they have how much acetyl-CoA is present, and eventually on all 3 lines things level off, okay? But the question is just a graph question. It says the maximum production rate of acetyl-CoA under condition 1, so that's your solid line, okay, is closest to which of the following? Now, the solid line kind of maxes out, so the maximum production, we're going to say around about here, okay, so on average, all right in this zone. Now, that's going to tempt a lot of you to put 65 micromoles per second. But that would be wrong, because if you look at the answer choices are rates, so a slope. It's micromole per second, micromole per second, micromole per second. So slope is rate, okay? So if they're asking for the rate of change or things like that, they're asking for slope, and it's going to be your rise over your run, which is your y value divided by your x value. So everything on this line, there are points. So there's a point there, there's a point there, there's a point there, there's a point there, there's a point there. And this looks very linear, okay? And anywhere we look at it, it's going to be about a 1-to-1 ratio, 20 over 20, 30 over 30, 40 over 40, 50 over 50, 60 over 60 is kind of what you're looking at. And that's where it maxes out. And then it just kind of stays there. So the trick question here is the maximum rate, okay? So rate is slope. Slope is rise over run. And so the maximum rate at this one is actually A, okay? So pay very close attention to what they're asking you for. All right, next question, number 4. Okay, zoom out a little bit. Okay, so you should have these in front of you, and it's very important. This is how they're going to do the questions. They're going to give you a scenario. Some of it we've talked about, some of it we have not talked about, and they could have asked a million questions about this, and I'm going to ask you some more questions about this specific experiment. But the one question they wanted to know is, which of the following statements best justifies the inclusion of test tube 5, so test tube 5 I have right here, as a control in this experiment? All right, so they're trying to measure, if you look at what they're investigating, they're trying to investigate the effect of the pH on amylase. Amylase is an enzyme. Enzymes are proteins, okay? And if you notice, like here, the pH, this one was at 7. We know that 7 is neutral. All right, starch added. Okay, they all have the starch added, amylase added, iodine added, and they'll tell you that iodine changes color when the amylase is present. But look at the one thing that is different about test tube 7 is it was pre-treated by boiling. Now, what you need to remember is that when we heat up enzymes, we heat up proteins, we do things like that, this denatures your enzyme. So because this was the one thing that was different about test tube 5, because it had other things in common with some of the other test tubes, then that's going to denature the enzyme. So that's what makes it the control, because it is the one where your enzyme activity is absent. And so that's going to be C. So it'll show you what happens when the enzyme is not working, okay? So the answer to number 4 was C. All right, number 5 is your good old U-shaped tube, and they said the scientist is doing an experiment. He has an artificial membrane, so that little membrane would be down the middle of our U-shaped tube. It mimics a phospholipid bilayer, so it mimics, and we know that means selectively permeable. He divides it, he fills distilled water, and fills both sides with distilled water. So there's water on both sides. And he puts a known amount of protein into the water on one side of the membrane. So I'm just going to put it over here. Doesn't matter, okay? And sometimes you need to draw pictures for yourself so you can see what the heck is going on. All right, so after some time, it doesn't say how much, the scientist measures the concentration of the protein on either side of the membrane, but finds out there's no change. So automatically we know nothing moved. So it probably didn't move because it couldn't pass through the membrane. Because typically things are going to move from high to low concentration. So they would naturally move if they could go through. All right, so it says, which of the following experimental changes, so now it's saying you're the scientist, would allow the scientist to observe transport of the solute across that membrane? So what does he need to change in his experimental setup to make sure he can actually see something pass through? All right, so this is your decoy answer, D. Add artificial aquaporins to the membrane. The aquaporins will allow something to move, but what do aquaporins allow to move? Water. Okay, so aquaporins would only allow water, and he wants to see a solute get transported. All right, so the best answer here is that he needs to use something that's small and nonpolar, instead of a protein, so that it can pass straight through the membrane. Proteins do not pass straight through membranes because they are not allowed to go through. Usually they're way too big. Okay, small, nonpolar, boom, pass right through. All right, so number 5's answer was B. All right, number 6. This is another experiment. A researcher claims that there's some factors that influence the function of certain enzymes. He sets up this whole experiment. He has 4 petri dish with bacteria strain 1 and 4 petri dish with bacteria strain 2. He uses a buffer, and if you remember, a buffer keeps it at a constant pH. And then he adds some sort of cellular contents to most of these. Okay, so 1 and 5 are missing those. All right, the substrate, 2 and 6 are missing it. And then the enzyme added. Okay, so 3 and 7 do not get the enzyme. So the question was, the design of the researcher's experiment is presented here. All right, so it says, which of the following statements best justifies, okay, so you're justifying why he included test tube 3 and 7. All right, so 7 and 3 are the ones without the enzyme. All right, so you had to go up to the table and figure that out, and there were lots of questions about this table. And the justification is A. They will show whether the isolated cell contents have enzyme activity or not. Because if you don't add the enzymes and then something still happens, then you know that there were enzymes already present in those cells. Okay? So answer number 6, A. All right, number 7. 7 had 3 questions that went with it. So they gave you this table, and they're showing bacteria, different pHs, changing over time based on, let's see, the treatment group. So we had the control, so they did nothing to it, and then the treatment group. And then they were looking at the pH. And if you notice on the table, the pH in the control group slightly goes more and more basic. And then in the treatment group, also slightly more basic, okay? But it gets more basic in the very end in your treatment group. And remember that that's from a 8 to a 7 is 10 times more basic or more acidic depending on which way you're going. So that's a big change, and tiny little changes can cause big problems. All right, so it says, which of the following was the dependent variable in this experiment? So your dependent variable is what you're measuring. And so what were we measuring? What will we be graphing? What will we be putting on our y-axis is going to be the pH. So number 7's answer is B. Now, still based on this set of data, they gave you 4 options of graphs. And the correct graph, because we have a change over time, should have been B. Okay, so there's B. And it shows a change over time, and it shows your control group and your treatment group, and it has them plotted. All right, this could be something very likely that they ask.